Welcome back, students. Moving on to a new week here and a new topic. It's that of grafting and budding. We'll have a topic on grafting today and again on Thursday, and that will be the last of the material for Exam 2, which of course is next week. And I'll send out an email reminder about Exam 2 next week. Principles of grafting and budding. This is something that's been going on since the beginning of civilization. Fruit and nut trees have been grafted because of the difficulty with getting some cuttings to root, and also because of the superiority that's achieved with high-value grafted crops. Grafting is among the most expensive propagation techniques, and even surpassing tissue culture. Budding, which is a variation of grafting, is even more expensive. If we look at the southeastern United States and think about high temperatures and the fact that we occasionally have flooding going on and wet soils, lack of oxygen in the soils, this creates a demand for, especially in plants, trees like birch, fir, oak, to graft them onto better adapted rootstocks. So therein lies an advantage. We'll talk about superior scion pieces, improved fruiting characteristics and taking advantage of that. Also there's greater reliance on integrated pest management in agriculture. We have reduced availability of pesticides and fumigants. And if you think about some of the root-borne diseases and insects of the root system of plants, well, we're doing something. We're breeding plants that have disease-resistant roots, or maybe some insect-resistant roots, and we're using those as rootstocks and grafting on superior scions to reduce our reliance on chemical pesticides. So, as we think about grafting, I mentioned that it's been around since the beginning of civilization. There's evidence that the Chinese were doing this going back to 1500 BC. If you think back to Greek civilization, ancient Greece, Aristotle, the great Greek philosopher and writer, in some of his writings about 340 BC, explained the technique of grafting rather well. And one of his students, Theophrastus, about that same time, wrote about grafting in great detail also. In the days of the Roman Empire, Paul wrote a letter to the Romans talking about grafting the wild olive to the tame olive, and he talked in such a way that his audience there in the Mediterranean region knew exactly what he was talking about. And that's true. The ancients were well aware of tree grafting and its importance to their food supply. And as we move on through history, we see record of grafting throughout the Renaissance age also. And it continues today in the modern nursery system in both fruit and nut trees and some of our shade trees. And it's beginning to be more accepted also in forestry crop production. So moving on here, some terminology. Let's just say grafting is the art of joining two pieces of living tissue together that will unite and grow into one composite plant. Yeah, it's just this art and science of joining two pieces together that grow together to form one single plant. So that may be a rootstock and a scion that are somewhat different from each other, or maybe they're quite similar to each other, but they're two different plant parts. Budding is a form of grafting where the scion is reduced in size. And of course, we noted this last week in lab in the T-budding technique on those fruit trees. Rather than using a scion that was an entire stick, we used a reduced scion that was a single bud. So we join these two plant parts together, the scion and the rootstock, the scion being the top portion of the graft combination, and of course the rootstock being the lower part of that graft combination. And if they heal, we continue to train that tree into a mature, well-branched, well-shaped tree that yields fruits, nuts, or provides shade or some other ornamental value. So maybe think of it like this. That graft, the scion piece, grows and is trained sequentially year after year with selective removal of branches to become the top portion of that grafted tree. And of course, the rootstock remains as the root system. So this isn't a mixing of genetics. We're not crossing two plants. Maybe the analogy is like a heart transplant. Someone goes into the hospital, their heart has stopped functioning, and they're fortunate enough to get a heart transplant, and somebody else's heart goes into their body. The doctor takes care of them. They're put on some medication. They heal. They continue to live. And it's not a mixing of genetics. It's somebody else's heart that functions along with that person, and that person continues to live. And so it's the same with this tree. There's not any mixing of genetics. You have two separate genetic lines here with the rootstock and the scion. Some other terminology that might come up as you discuss or read about grafting, and one might be the interstock. So as noted, the top portion is the scion, the bottom portion is the rootstock. Occasionally, those two pieces aren't compatible. They're similar but not compatible. They won't heal to form one composite plant. So there are instances where we use a third piece, and it goes in between the scion and rootstock. Of course, it has to be compatible. It has to heal with the top portion and the lower portion. Nonetheless, it all heals together, and we have one composite plant. Of course, if there's an interstock used, it has three pieces. All right, some other terminology. Recall vascular cambium. This is a very thin layer of cells beneath the bark, but to the outside of the wood. Okay, so to the inside would be all that xylem tissue and dead wood, and to the outside would be the phloem and the bark. Vascular cambium. It's also a meristematic region. That tells me that those cells are parenchyma cells, and they're capable of dividing and reorganizing and forming other tissues. Also, callus tissue. Callus is composed of parenchyma cells. It tends to be a disorganized group of parenchyma cells that's required to develop for success in graft union formation. Now, as you recall, last week we were talking about callus formation in the rooting of cuttings, and we said callus tissue isn't necessarily important or even needed for root formation. As we graft two plant parts together, it is central to our success, this callus formation at the union of those two pieces. And so it is cells in that vascular cambium after the wounding process that begin to divide and further divide in both the scion and rootstock, and they begin to intermingle into this disorganized mass of parenchyma cells. That would be the callus tissue. All right, so to further recognize these tissues, again, the vascular cambium is this very thin layer of cells that you can't see without the aid of some magnifying device. It's only 2 or 3 cell layers thick, and again, it's to the outside of that secondary xylem, but it's to the inside of the phloem tissue. Okay, so to the outside of that vascular cambium, collectively we refer to that as the bark. To the inside, we refer to that when grafting as the wood. And the premise here is the scion and rootstock, we place them together, and what's key to success here is the vascular cambium from both pieces needs to be touching or in very close proximity to each other. So in the first stage here, we're lining up our scion and rootstock. In some techniques, it's important to have two pieces of a rootstock and scion that are identical width to ensure that those vascular cambiums are in very close proximity to each other. And so through this wounding process, that meristematic region, those cells begin to divide and divide, giving rise to new parenchyma cells. They intermingle with each other. And so this takes a period of time, maybe up to 14 days. And then beyond that, there's still more callus production, but some of those newly formed parenchyma cells begin to organize and become specialized. Some of them become phloem cells, others become xylem cells. And this leads to the formation of a vascular bridge between those two pieces. And if the healing is successful, we have a successful vascular bridge now between the rootstock and scion. Again, remember that scion piece doesn't have a root system, very susceptible to desiccation. But as soon as that vascular system is now established, it has a pathway for water to be translocated into it. And over time, that phloem has usefulness also, of course, as it translocates photosynthates from the scion portion down into the roots. So a brief introduction here to the steps in grafting that lead to a successful healing process and the formation of one composite plant. Lining up the cambium, cells in that vascular cambium go through cell divisions, giving rise to new parenchyma cells that form callus. Some of those cells becoming specialized into phloem and xylem. Eventually, a vascular bridge is formed between those two pieces. So as we move on here now and describe, we'll talk about the reasons that we graft this somewhat sophisticated propagation technique. I've already told you that it's much more expensive than cutting technology and certainly more expensive than using seeds. So yes, we'll describe why we might choose to graft. But first, let's focus on the rootstocks and how we come about growing rootstocks. There's really two methods of rootstock systems. One would be those rootstocks that are planted from seeds, and they grow up in containers or out in the field, either way. A seedling rootstock system, just planting seeds, it germinates up, grows for a year or two, and that becomes the rootstock. Or clonal rootstocks, whereby we're taking cuttings, rooting those cuttings, and then that rooted cutting becomes our rootstock. So for the nurseryman, he has these two options at his or her disposal, either starting with rootstocks from seed or with clones. Okay, so seedling rootstocks, they have their advantages. You know, seeds, you're just planting the seed, it germinates, it grows, you care for it, that's easier to manage. It tends to be much less expensive. We can mass produce them rather easily compared to mass producing cuttings. I think now you've had experience in both taking cuttings and also growing plants from seeds. And it's just much easier to grow plants and establish them from seed than with cuttings. Much less effort, much less management. So yeah, we can mass produce them. Maybe another reason the nurseryman chooses to use seedling rootstocks is we start with that seed. It's extracted from the fruit. Might go through a period of stratification, then we plant it. We're almost certain that there's no viral transmission from the mother plant into the seed. Okay, so this is a concern with some of our fruit crops, whereby trees can potentially be infected with viruses, and some of these viruses don't show any symptoms immediately. Even though the entire tree would be infected with the virus, the entire tree could not show any symptoms. And if I use that tree for cuttings, yeah, my cuttings would have the virus also. And as those cuttings grew and mature, those fruits might have that virus also. But I can get around that by using seed. The mother plant, if infected with a virus that causes damage, that virus doesn't get passed off to the seeds in that fruit. So this is a potential advantage as nurserymen consider their rootstock systems. So as I said, we take the seed, we can plant it in containers or maybe out in the field. All in all, when they mature, they tend to be much deeper rooted, well established, compared to those that were established from cuttings. And that has advantages certainly after grafting and placement into the orchard, having a strong root system on a tree so that it goes through the transplant process with as little shock as possible. And that just encourages a stronger tree to be established in that orchard. So that would be some of the advantages of using seedlings as rootstocks. And if we compare that to clonal rootstocks, they've got some advantages also here that we need to mention. Certainly, one is the uniformity of a clone. Remember that with a clonal rootstock, as the name implies, it's a clone. So if I have superior root characteristics in that plant, all those cuttings will have the same genetics, the same characteristics, including that superior rooting characteristic. So uniformity. Is this always true in seedlings? No. In some species, the seedlings exhibit so much heterozygosity that, wow, some of the seed might have good root characteristics, others may lack that trait, going back to the fact that that seed arose from meiosis and the fertilization step, that recombination of genes. Of course, that doesn't happen in clonal rootstocks. I'm taking cuttings, and they are all clones. It is possible with some crops to influence the final mature size of the tree or growth habit based on that clonal rootstock. We see this in apple production. We have apple rootstocks that are clones, and some of them are vigorous rootstocks, allowing the apple tree to grow up to 25-30 feet in height. We have less vigorous rootstocks that control the height of the tree and only allow it to finish growing at about 12 or 15 feet in height. And we have some super dwarf apple rootstocks that we could graft on that same apple variety to it, and because it's a dwarf rootstock, the tree only grows to 3 foot tall. So yeah, that might be the advantage of using some clonal rootstocks, is that they control the overall size of the mature tree. The second part of this bullet, growth habit of the grafted plant, we see this in some ornamental plants where it has a weeping form. And that may be an advantage if we want to have the weeping form like a weeping cherry tree or a weeping red bud. So that's potential also. So now by and far, as we look globally at fruit production, apple rootstocks, pear rootstocks, plum, cherry, grapes, these are some examples of plants that are commonly grafted onto clonal rootstocks for some of those reasons that we just described. So again, the nurseryman has at his or her disposal an option with rootstocks to go with seedlings or with clones, and they both have advantages. The system needs to be thought out thoroughly and a sound decision made. And again, sometimes it is because of the specific species I'm growing. If I'm going to grow apple trees, without a doubt, I'm going to use clonal rootstocks. If it's a crop like peach, I've got some choices between seedling rootstocks and clonal rootstocks. So let's move on to some of the reasons why we may choose to perform this very sophisticated propagation technique, grafting, or its variation, budding. We might be trying to perpetuate a clone that can't be propagated by some other asexual means. We found a plant that has some superior fruiting characteristic. It tastes good or smells good or combination, higher vitamin C, whatever trait it has that has value to us. Perhaps it has to be cloned because there's too much heterozygosity in the seed. In other words, too much variability in all the seeds, so it requires it to be cloned. At the same time, maybe cuttings don't root easily, or it might root easily, but the root system just doesn't perform well in most of our soils. So there we may choose to perform grafting. So again, perpetuating a clone, and with the benefits of certain scions, again, coming back to the fact, we do this all the time in fruit tree production and nut production, whereby we breed different parent plants, evaluate the progeny for, quite commonly pecan. One of the top priorities is pecan scab resistance. And another top priority in pecan breeding is, thin shell. And so that we may have a new variety that has both those characteristics but doesn't have the best root system. Nonetheless, we've selected out a plant for superior fruiting characteristics, and we see the same in peach, plum, apricot, apple, and we could go on and on with the benefits of certain scions. And again, those plants may not have well-adapted root systems for all the potential soil types and climatic conditions, pest pressures. So therein lies the benefits of certain rootstocks. And yes, there are breeding programs in place to breed improved rootstocks from tropical fruits to temperate fruits. Even in our nut species, we attempt to improve our rootstocks. Rootstocks that grow with greater vigor and establish more rapidly for more rapid maturity of the tree and faster production, or maybe, as we mentioned, it's disease resistance or the ability to tolerate more alkaline soil conditions. So we can maybe summarize it like this. Those rootstocks may have greater resistance to environmental stresses like soil salts, or maybe it's a heavy clay soil that it's tolerant of, or perhaps some soil-borne disease. We also mentioned the fact that rootstock may have a benefit that it controls the mature size of the tree. If I'm an apple grower in the Pacific Northwest of the United States, there's advantages to having trees that are maybe only 6 to 8 feet tall versus trees that are 30 feet tall. Safety of the workers as they're picking them, or just, maybe I don't have mechanization for harvesting and they have to be hand harvested, therefore I want to keep shorter trees. So this is part of my decision making as I choose rootstocks for grafted trees. There is some reporting also that with some rootstocks and grafting onto them, it hastens the reproductive maturity. In other words, the grafted plant enters flowering and fruiting sooner. Fewer years required for maturity. And of course, that has an economic advantage to that orchard manager. So maybe hastening reproductive maturity. In some instances, we see advantages to transplant success being enhanced. This has been shown in some of our shade trees, from maple to Japanese maples, where selected rootstocks better withstand some of the stresses of transplanting into the landscape, but the top part has been grafted with a scion that has superior foliage characteristics. So again, here to summarize the benefits of specific rootstocks. Controlling size, we talked about how in apple that we have numerous apple rootstocks that are available to us. They come to us from the Malling-Merton Agricultural Research Station in England, where they've done a tremendous amount of breeding and research in apples. And this is an agricultural experiment station that's been in existence for, I'd have to check back, 160 to 170 years, and they've got excellent records, several crops that they've researched over time, and they've released the series of apple rootstock clones that are rated from vigorous all the way down to subdwarf. So that, the vigorous rootstocks allow that tree to, I could have the same scion grafted to each of these rated categories of rootstock. Let's say they all are grafted with Golden Delicious. The vigorous rootstock will allow that Golden Delicious apple tree to grow up to 25, 30 feet in height. Whereas I can also take that Golden Delicious, graft it onto the subdwarf rootstock, and it will only mature at about 3 or 4 feet in height. So regardless of the rootstock, it's still going to yield the same type of apple, Golden Delicious. They'll all taste the same. The fruit will look the same. The difference will be the final mature height of the tree. Now you may be thinking, wow, there's probably far fewer fruits on those subdwarfs. That is true. Many times those super dwarf fruit trees are more for homeowners. They will plant a few of them in the yard or maybe even in large containers on their patio. Not too standard for orchard use. Much more common in the orchard would be the vigorous down to the semi-dwarf. All right, so plant breeders will, to our benefit, improve our food supply by breeding better fruits, better nuts, and that also includes breeding better rootstocks. Coming back to interstocks, remember we said this was the third piece because perhaps the rootstock and scion aren't compatible. And so one example here is the plant on the left is quince, Q-U-I-N-C-E, Q-U-I-N-C-E, quince. And in this instance, it is the interstock between Bartlett pear being the scion piece, and Old Home being the rootstock of this pear tree. And we're using this quince as an interstock. And of course, this tree is matured, it's transplanted out into the orchard, and will continue to mature into a productive pear tree. Okay, so we've described benefits of scions, benefits of rootstocks. Moving on with other reasons that we might graft. There is a technique known as top working. Top working is grafting trees with scions onto established trees in the orchard. I'm grafting onto established trees in the orchard, or it can be in the home landscape also. So I still have a scion and rootstock. The difference is this rootstock is already in place. It's at its home. It's typically in the orchard or maybe in your front yard, wherever. Why would you want to do this? Top work your trees. It's an advantage of changing cultivars. So, as an example, perhaps an apple orchard manager has a couple hundred acres of apples, and it's a specific variety that doesn't get a very high price in the marketplace, in fresh market. And he doesn't have access to the frozen market or the juice market. And there's new varieties coming out that take a higher price at the marketplace. And you've probably seen this when you go to the grocery store. There's variation in price per pound of apples. You know, the Macintosh may be 69 cents a pound, Red Delicious 99 cents a pound, Envy $1.69 a pound. And so as I start growing apples, if I have one of those varieties that doesn't yield as much in terms of dollars, I might change my orchard to a newer variety that yields more in terms of dollars. Typically, you don't do this all in one season. You'd have to set aside sections of the orchard, maybe spread it out over 4 years or 3 years. And, you know, if I decided to do this top working over 3 seasons, the first season I would cut and graft a third of the orchard. The next year, another third, maybe wait a couple years and do the last third of the orchard, so that I don't take the entire orchard out of production in one season. So we see this occasionally in fruit and nut tree production, where the grower wants to change cultivars. Again, another reason for grafting. And we describe this as top working. And, you know, again, it's not just limited to the orchard manager. Home gardeners can perform this technique, you know, changing cultivars, adding multiple cultivars per tree. Some other reasons to graft. This isn't common at all, but repairing the tree because it's been damaged some way. So this might be in an orchard, or maybe it's your favorite fruit tree in your backyard, and maybe a beaver comes into the yard and gnaws on it and damages it. You know, he's trying to get that tree to make his house, but you know, maybe you are able to keep the beaver away, and you want to fix the damage to this tree. And I say that example because I know somebody that happened to with a plum tree on Lake Buchanan in central Texas. Or maybe the damage is from a tractor hitting it or an implement. And it's damaged that side of the tree. Thereby, there's no more xylem and phloem flow. The flow of water and nutrients in the xylem, the flow of photosynthates through the phloem is now eliminated on that side of the tree because of the damage. Therefore, that side of the tree in the canopy, it's quite possible those limbs, that section of the tree is going to die back, and likewise with the root system, it could decline also. With that decline, the tree could eventually be weakened and potentially die. So if the tree has value to me, it is possible to repair it and improve its health. So to do this, above and below the damage, we make cuts into the bark. Then we go up into the canopy and we remove some of the limbs from the tree, remove the leaves, and we insert, you have to remember polarity here, so that the distal end of that branch goes above the damage and the proximal end goes below the damage. They've got some grafting tar that they put on there that seals it off to keep it from losing water, to keep insects and diseases out. And of course, that encourages the healing process. And if they all heal properly, it reestablishes a vascular connection on this side of the tree. And of course, as that tree grows in girth each year, you know, it's getting a little bit wider year after year. Likewise, those branches that are grafted on to that portion of the tree, they too will grow in girth season after season. So this bridge grafting or inarching technique of repairing damage to trees of value. Okay, some other reasons to potentially graft trees is what we say is virus indexing. So there are instances where plants become infected with viruses. Sometimes that virus, we see the symptoms immediately in the foliage, in the fruit. It may cause damage to the tree, may cause it to die, or may cause reduced production if it's a fruit tree. But then there's also viruses that when they infect the plant and travel throughout the cells of that tree, they don't show the symptoms immediately. And so the value here might be in that, well, we've got all these rootstocks, and are we certain that they're virus-free? They don't show any visual symptoms, so should we just continue to hold on to them and grow them for 2 more years and hope we see some visual symptoms, then we can rogue out the ones that are infected? Well, what we can do is graft on to some of those rootstocks an indicator plant. This has been shown to be possible with cherry. Cherry, of course, has some specific rootstocks. Then you have some other specific scions for the grafted tree. And that indicator graft is, well, what will happen is I can graft onto some of those rootstocks, like T-bud them, and if it begins to ooze and discolor after about 3 or 4 weeks, it's an indicator that that tree is infected with a virus. Now I suddenly have visual symptoms of the virus in that tree, and I can get rid of it. And of course, the trees that don't exhibit the ooze at the point of the budding, yeah, I know it's healthy, it's free of the virus, and I can go on. So that's one approach to what we say is virus indexing. Another would be whereby we might have our stock plants infected, or maybe it's even the scion of choice infected with a virus, and use of seed is not an option. It has to be vegetative propagation. Think about the growing tip of a plant and how plants grow. So in this graphic, you'll see in the left side, there's a branch. It's in the wintertime, it's leafless, and a magnifying glass to show us that terminal bud. And we magnify it, and we see the tip of that bud, where there's some very small leaves that have formed, and they're starting to emerge. But the very tip of that branch, the apical meristem, that dome, that apical dome, on the graphic it says apical meristem, but that dome right there, that is the youngest point on that branch. Those cells are rapidly dividing. They eventually, some of those cells will differentiate into leaves or the petiole of the leaf or xylem tissue. But that's a meristematic region, and those cells divide rapidly. Also, and this is important, at the tip of that apical meristem, it will be free, those cells will be free of that virus. That virus will not be in those cells of that apical meristem. Those cells are growing and dividing so quickly that the virus doesn't travel fast enough to get into those newly dividing cells. So maybe think of it this way with this next graphic. If I examine it even more closely, those cells that are denoted with red, those are infected with virus. And the green ones are healthy, they're uninfected. And at that apical dome, that's where those cell divisions are occurring rapidly. If I can somehow remove that portion, if I can just, get a very small razor blade and a really fine microscope, I can remove that apical dome, transfer it into tissue culture. So now think of the test tube plant. I've got this apical dome that I've removed, placed it onto agar, and I grow it out in tissue culture to the point where I now have a Scion piece that isn't infected with the virus. And so, think back to our field trip to Magnolia Gardens tissue culture lab. I don't think we saw this, but they were doing some of this virus indexing with both potato and blackberry. So again, one of the advantages of virus indexing. So we're trying to summarize here some of the purposes, some of the reasons for grafting. So if we think about the formation of that graft, coming back to these steps that are occurring sequentially to the point where we have one composite plant. We take the two pieces, we line them up together. What's central to our success here is the vascular cambium of both the rootstock and Scion need to be touching or very close proximity to each other. Again, our final goal is to have a vascular bridge that has formed. And if there's distance between the vascular cambium of the two pieces, it's not very likely that it's going to succeed. It's just going to lose energy and die before we form that vascular bridge to the Scion piece. Okay, so maybe we're doing it right. We have the right rootstock and Scion. We've carefully lined up the vascular cambiums. So we've got them in good, close, intimate contact. And that's important also, that they are in good contact with each other. And that requires some type of rubber band, tape, adhesive to hold it in place so there isn't any slippage or movement. There is a wounding response too. There's some cutting that went on and some of those cells were broken open. They dry out and become necrotic. So yeah, there is a wounding response that goes on, and those cells of the vascular cambium are stimulated through that wounding process to start dividing. Remember that vascular cambium, it is a meristematic region. Cells are capable of dividing, and those new cells eventually can mature and become specialized cells. Beyond that wounding response, there needs to be callus bridge formation. So both pieces, the vascular cambium gives rise to new parenchyma cells. Those cells intermingle with each other to form a lump of callus tissue. And eventually, specific cells differentiate into xylem. Other specific cells differentiate into phloem. And that continues until the two pieces are joined together, or we say they're bridged together. And that wraps up the last of our formation of the graft union. We have differentiated vascular tissue. Some of those plain old ordinary parenchyma cells that were callus, some of them differentiated into phloem, others differentiated into xylem. And so with that, I now have connective tissue bringing water out of the soil through the roots up into my Scion piece. And eventually that Scion piece will mature and grow, go through photosynthesis, and have sugars that it can translocate via the phloem through that vascular bridge down into the root system. So the steps behind the formation of a successful graft union. All right, so as we consider that, I think it's important also to evaluate other considerations that influence our success. What's going to influence our success? Are we going to be 100% successful if we graft trees all day? Or will we be 60% successful or 30% successful? Let's consider what's might be under our control or not under our control that we need to think about that influences our success. One might just be the plant species. Some plants, the success in grafting is just a lower percentage. They're much more difficult to form a vascular bridge. Some examples would be the hickories, beeches, oaks. They tend to be more difficult to graft, as opposed to plants like grapes, pears, and to some degree peach. Those are much easier to graft. So some of this success is just coming back to the plant species. Some other considerations we might take note of that influence our success. Remember, we're joining two plant pieces together. We want those cells to start growing, divide to form callus, eventual vascular bridge formation. So factors like temperature. But what temperature? What are some optimal temperatures? We don't have a lot of data on that. Cell divisions really slow down as the temperature approaches freezing. So at 32 degrees, you wouldn't expect too many cell divisions at all. On the other extreme, as you get into the low 100s Fahrenheit, there's not going to be much cell division going on. So optimally, it depends on the plant. There's been some measurements in apple that show that, if temperature's in this about 70 to 80 degrees Fahrenheit, that tends to be an optimal range for cell division and callus formation. So yeah, it depends on the species, but think about the temperatures and how that might influence cell division and the formation of the graft bridge. Certainly the moisture status of the plant. Those cells need to be fully hydrated. And remember, you're moving a piece of scion tissue. You need to take that from a healthy, hydrated tree with as little stress on it as possible. Same with the rootstock, just as little stress on it as possible. Other factors that might influence, environmental factors that influence our success, might be the growth activity of the rootstock. If I'm performing the T-bud, well, the bark needs to be slipping. That doesn't happen all the time. There's a specific period of time in the spring when the bark easily slips away from the wood, and we can perform techniques like T-budding. Now, there are some other budding techniques that we'll describe on Thursday where we can get around that and perform in the wintertime or maybe in the fall. All right, so that's another consideration there. What else? Coming back to polarity. So think about this. When you're joining these two plant parts together, the rootstock and scion, with my scion piece, the proximal end of the scion piece is joined onto the rootstock. That is the proper polarity, which of course leads to callus formation, vascular bridge formation. So think about polarity also. And this one's difficult to gauge, craftsmanship. It's probably the more you graft, the more experience you get, the greater your success. And the more frequently you perform grafting, the better you get at it. Maybe think of it like riding a bicycle or throwing a football. The more you do it, the better you get. And if you do it occasionally, you don't forget how to do it. And it's the same with grafting. The more you do it, you sharpen your skill, your techniques. And if you do it, year after year, you get better at it. So yeah, craftsmanship. I can recall having an internship when I was an undergraduate at a large nursery outside of San Antonio and spent an entire week T-budding Santa Rosa plum onto peach rootstocks. And it was really the first time I had done it extensively. And I was working with two guys that had grown up in apple orchards in Mexico and had grafted their entire life. And so, they're going back 3 or 4 weeks later and looking at our graphs, they had success rates on their grafting that was near 100%, and mine was a pretty low percentage. So again, it comes back to just craftsmanship. There's a certain technique. You don't want to let the tissue dry out as you transfer it over to the rootstock, the way you wrap it up. So this is part of influencing factors. Oh, maybe also competitors, insects, bacteria, fungi. We want to keep them away from that wounded tissue. So this can also, if I'm not careful, be detrimental to my success. Competitors like insects and bacteria and fungi. All right, folks, so these are the influencing factors in my success. We talked about the reasons why we may choose to graft versus some other propagation technique. And we'll continue on with this topic next Thursday and talk about some specific grafting techniques and look at some videos also of specific grafting techniques.